channel. Today we are going to learn about one of the very important topic, that is chromosomal mutation. We'll learn about all the different subtypes of chromosomal mutation and what is chromosomal mutation. Now before knowing about chromosomal mutation, you should know that chromosomal mutation is one of the major types of mutation. Mutation, it is any change in the DNA sequence of an organism. And there are two main types. And there are various different types and subtypes based on different characters, which we have already discussed in our previous video tutorial. So please check all the details, all the basic details about mutation which we have discussed in our previous video tutorial. Please check it in the description box below, because it is going to help a lot for your competitive exams. You must have a clear explanation about mutation, which we have already given in our previous video. So it is given in the link. Please make sure that you go through the link. And also subscribe onto our channel so that you don't miss out such important videos. These videos are made on very important basic topics of biology. So if you want to crack any competitive exam, you must have a thorough knowledge about all such basic topics, which we are covering in our channel, that is Science eSolutions. So make sure you subscribe onto our channel. So what is chromosomal mutation? Chromosomal mutation, it is a type of mutation which affects the structure of the chromosomes. So there are two major types of mutations. One is chromosomal mutation and another one is the genetic mutation. Okay? Genetic mutation, it affects the genes as the name suggests. And chromosomal mutation, it affects the structure of the chromosome. So in today's video, we will be thorough about what are chromosomal mutations and what are different types of it. Okay? In our next video, we will be checking the genetic mutation. So what is chromosomal mutation? Any mutation which affects the structure of the chromosome, that is called as chromosomal mutation. And this mutation is sometimes also called as large scale mutation. Large scale mutation, because it affects the chromosome, structure of the chromosome, and a chromosome has various genes in it. Many genes are affected. That is why it is a large scale mutation. Okay. Now, chromosomal mutation, there are 5 subtypes of chromosomal mutation. The first one is deletion. As the name suggests, a chromosomal segment is broken. Okay. There is a breakage in the segment of a chromosome. So first there is a breakage in the segment of the chromosome. It is very clear in this diagram that this one is break in the chromosome. This is the chromosome, and this red portion, it denotes breaks in the chromosome. So chromosome is broken. This part of the chromosome is broken, and this part, it is deleted. Okay. Deleted genetic material. This genetic material is deleted from the chromosome and the rest part is joined. So now the chromosome becomes shorter because it has lost one part due to breakage. Okay, and the rest part they rejoin and the chromosome becomes shorter and some of the genes are lost. Okay. So when the structure of the chromosome is broken and then rejoined, that is called as deletion, because part is deleted. Clear? It is clear from this diagram, I hope. So this red portion of the chromosome is broken and all the genes which were present in this red portion, they were broken down, and the final chromosome, it is like this. So all the genes which perform different functions, they will not perform because the genes is lost. Clear? Now the second type is inversion. Okay? Inversion. What happens in inversion is first deletion, same as the first one. One segment, one part of the chromosome is broken down. You can see here this red and green part, it is broken down. Okay, this part it is breaks in the chromosome. Now after it breaks, then there is a flip. That means the lower one becomes the upper part and the upper side goes to the lower side, right? Here you can see green was in the lower one and red one was above. Now it is inverted. Green becomes the above part and red becomes the lower part. Okay. So now you see there is a break and then the segment flips. The broken down the segment, it flips off. The segment, it flips off. Okay, upside becomes down and downside becomes up. It flips and then rejoins. So the sequence of the genes is changed. Sequence of the bases is changed and hence genes are affected and the chromosome is affected. It is not present in that structure where it should have been. Okay. So this is inversion. First break in the chromosome, you can see this part of the chromosome is broken down and then the segment flips, turns around, okay, it flips and then again rejoin, or you can say reinserted DNA piece which is flipped and inserted. Okay. Now the genes, the sequence of the genes, sequence of the bases is changed. So such type of chromosomal mutation where there is a break and then flipping off of the segment, that is called as inversion. Hope these two subtypes of chromosomal mutation, that is deletion and inversion, is clear to you. Students, if you understand the concept which we are going to provide you through this video, please give us a like because it motivates us a lot to create such educational videos. So let's move on to the third type, that is duplication. In duplication, a segment is doubled, duplicated. One segment like A, B, C, D genes are present, they are again duplicated. Means due to some problem during meiosis, that segment is again repeated. So A, B, C, D genes which were formed, again it will form A, B, C, D. That means it forms twice. Over means presentation of the genes. The genes are present over many in numbers. So this is duplication. You can see this segment, it is of this length, it is duplicated and becomes double and then again rejoined. So this is called as duplication where the segment is duplicated, becomes double, okay, due to some problem during meiosis. Then come to the fourth type, that is translocation. This translocation, it involves two different chromosomes which are non-homologous. Okay, we will be taking two different chromosomes which are non-homologous, that is chromosome A and chromosome B. You can see chromosome A blue in color and chromosome B it is filled with red in color. Okay. Now see, before translocation they are like this in structure. Then after translocation what happens? There is a break in this part of A and this part of B. Now the segment is interchanged between A and B. And remember, A and B chromosomes are non-homologous chromosomes. Okay. So this B, this part of the B, this red part of the B, this one, it comes here in the A chromosome, and this blue part of A chromosome, it comes in this part of the B chromosome. So there is segment interchange between the segment of the non-homologous chromosomes. Okay, non-homologous chromosomes, they interchange their segments and this is called as translocation. So this is before translocation and this is structure is after translocation. Clear? So this is about the fourth type, translocation. I hope this is clear by these diagrams. Then the fifth one is non-disjunction chromosome. As the name suggests, non-disjunction means there is no separation of chromosomes during meiosis. We know during mitosis or meiosis, there is a proper separation of the chromosome so that the chromosome number is retained. Now, in non-disjunction type of chromosomal mutations, due to some problems, what happens due to some errors or mistakes, the non-disjunction, the separation of the chromosomes is not proper. So sometimes many chromosomes are present in the egg cells. Okay, like or sometimes less number of chromosomes are present. Like you can see this one, Down syndrome. Down syndrome, there is 21 trisomy. In the 21st pair of our chromosome, there should be 2 X chromosome. But the person who is having Down syndrome, there is 21 trisomy, or 21st chromosome is triple X. Okay. Then similarly, there is Klinefelter syndrome, Turner's syndrome. Turner's syndrome. This is XO. It is having less number of, the person is having 1 chromosome less in their X's, or in their final, this sex chromosomes is having 1 less number of 1 X chromosome. And again Klinefelter syndrome. Klinefelter syndrome. It is XXY. Okay. So the final sex chromosome, it is having XX and Y. So this is clear that due to non-disjunction, non-segregation of chromosomes during meiosis, the chromosome number is affected. That is why it is called as non-disjunction, non-separation. It means non-separation, or there is inability to separate properly during meiosis. So these are the main basic 5 types of chromosomal mutation: deletion, inversion, duplication, translocation, and non-disjunction. Hope you have understood this.